What if a bionic hand, in addition to moving with your thoughts, could also let you feel the edges, textures, and even the motion of objects sliding across your skin? In this special episode of the Neurocareers BCI Award Series, we sit down with Dr. Giacomo Valle, the first place winner of the 2024 BCI Award, to explore a groundbreaking leap in neurotechnology, synthetic touch for brain-controlled bionic hands. Together with his team, Giacomo has advanced the frontier of tactile feedback by using intracortical microstimulation ICMS of the somatosensory cortex to restore the missing sense of touch. By delivering spatially and temporally patterned electrical stimulation, they have been able to evoke sensations of edges, tactile shapes, and motion across the skin, allowing individuals with paralysis to experience a more natural, dexterous, and meaningful connection to the world through their brain-controlled prosthetic hands. This innovation is a glimpse into a future where prosthetic limbs both restore function and bring back the feeling of being human. So stay with us as we dive into Giacomo's research journey, the science behind patterned microstimulation, and what this breakthrough means for the future of BCIs and neuroprosthetics. Oh, welcome, Giacomo. It's a great pleasure to have you again on our podcast.
Yeah, thank you. Thank you very much, Milena, for the invitation again. And it's a pleasure to be here and update on new studies, on new work, and about the neuroprosthetics. So very excited.
Yes, thank you so much for being here again. And where are you joining us this time? I remember you were joining us last time from Sweden. So where are you located at the moment?
Yeah, I'm still in Sweden, so I can confirm that now I'm more stable, like in terms of location in the world. So now I'm assistant professor in bionics at Chalmers University of Technology in Gothenburg, and with my team we study how to restore sensory motor function through the use of neuroprosthetics and BCI in people with different type of neurological impairments and affecting, of course, the sensory motor system. Yes, I collaborate a lot with Chicago still, so with Chicago and Pittsburgh, so with the Cortical Bionic research group, where actually also the study was performed and the study that won the BCI Award 2024. So we still keep the collaboration. We still have studies and research together. It's very fruitful, very, very nice to continue to work with these BCI participants.
Yes, very, very interesting to get more details about that. And can you tell us about this project, yes, that won the first place this year in the BCI competition?
Yeah. I'm very proud of this work. Firstly, I want to say that the work was actually performed at the University of Chicago in the lab of Sliman Besmaya, who passed in 2023 in August. And this is a kind of the last study that is also somehow celebrate his career in neuroprosthetics as well. I feel in BCI because was the part that was combining the neuroscience and the study of touch with the application, the translation application of this knowledge, leverage this knowledge to develop technology able to restore touch. And actually the study was published in Science and also got this prestigious award. So I'm very, very happy also for Sliman as well to have this milestone achieved with this last big study. So this is where I want to also mention this because it's something that we did together as well. So he was present during the first part of the study. And then I continue with Charles and Rob and the other members of the team. So the study is actually, as you know now, because you know me a little bit from the previous podcast events and meetings, I started from the periphery. So I moved in this field more recently, like in the last 3-4 years, trying to see how to restore the sense of touch and sensation in not anymore in prosthetics for amputees, but in a field where people with paralysis, they're also suffering from a deferentation, so they don't have any more connection with their body, even if their body is still present compared to amputees where actually you don't have the body part. Here you have the body part, but you don't actually feel and move this limb. And actually there are two approaches. One is trying to regain function of the native limb, reactivate with functional electrical stimulation, exoskeletons. And then there is this other approach that we were trying to follow, is to have an extracorporeal limb, so a prosthetic device attached to the wheelchair or on the table that you can control with your thoughts and with your volitional control, but you can also sense from the sensors that you have on the prosthetic. So the concept is at the end kind of similar to the amputees condition, no? Where actually you want to again do that, but instead of recording from the muscle, in this case, we record from the brain. And instead of stimulating the nerves, here we stimulate in the cortex. So this was the first time that I was approaching this problem, and I transferred my knowledge from the periphery more on the cortex. So this is how the study started, trying to restore that. And also there is a lot of, we can maybe discuss on this, that we took the inspiration from the visocortical prosthetics and what was achieved in trying to restore vision in blind individuals, stimulating the visual cortex instead of a somatosensory. So we also took a lot of inspiration from their results.
Yes, yes, so many things to discuss. And I truly appreciate you acknowledging the team that you worked with and Professor Bismaya, who, like you said, passed away. And I think I saw how big was that loss for the whole community, and there were so many posts, and people really were taking it very close to the heart. For those who maybe are not very familiar with Professor and his work, can you just give us a brief overview of his contributions? What did he did to the field and what he was known for and still is known for?
Yes, yeah, thank you. So we are still following his footsteps, no, and his steps in the field with what he discovered. Because Sleeman was probably one of the most famous and acknowledged neuroscientists in the field of somatosensation and touch. So he started to study this in non-human primates for many years, doing neural recording in the periphery, in the spinal cord, in the cuneate nucleus, in the thalamus, in the cortex, different part of the cortex involved in the sense of touch. So he was a neuroscientist, so he really was interested in understanding the mechanism behind our experience and perception of touch, so understanding how the brain is encoding this information, how the fibers, the different sensory channels. So he has a long history of researches on that. So starting from motion, edges, indentation, pressure, and all these type of tactile features that we experience when we touch something. Our sensory afferents and cortical neurons are coding these features in different ways, and he was trying to disentangle the contribution of that in this multidimensional encoding process. Then he also contributed a lot in the field of psychophysics, where actually he was studying this not only in human primates, but also at the perceptual level with healthy individuals. So how we perceive textures, how we perceive motion, how we perceive orientation with our hands when we manipulate objects passively or actively. So this is another layer. So we have the neural coding with the non-human primates, and you study this with implants, with neuroprosthetic devices. And psychophysics, you can study the natural perception of health individuals, how you actually feel these features and how you rate this in terms of naturalness, in terms of intensity. So this type of field. And then in the last part of his career, he started to translate and to be the reference person for the BCI, aiming to restore touch, because he was one of the greatest experts in this. And so neuroengineers ask him to help in how to design an artificial sense of touch. So he was the first one contributing with the first implant of this kind in the somatosensory cortex. And the Fleischer et al. Paper in 2016, they published the first work with a spinal cord individual was implanted with the array in the sensory cortex. And through the stimulation, they were able to restore the sense of touch, stimulating the brain in a precise way. All his work is also very important for the BCI in terms of safety parameters that we can use for restoring touch in humans, because he did these very seminal studies on how much you can stimulate the cortex without damaging the cortex. So he did this in animal. He published this, that is safe to stimulate with this parameter, and still in the FDA dossier of our BCI, there are these studies as a reference for the parameters that we use. And these are very important because we can use this range because of his studies. So in many different aspects, he contributed from the basic neuroscience to the psychophysics, and then for the BCI for the first steps. And until this study, that was the last one in the field. At the end, it is a kind of mix of the two, no? Because we have edge and motion, and the part of the fine features of touch and the BCI.
Yes, yes, and definitely his work will continue because it's in the essence of all of it. How was it like to work with him?
Exactly. Then there is all the other part where actually he was a rock star in everything, like from the how approach life and approach science, so very with curiosity-driven approach, with passion, really able to transfer this passion to the entire lab, to the group. And this is the reason why everybody was really trying to do his or her best to for this research. Motivate also the BCI participants, because if you have the opportunity to talk with our participants, they were really motivated also by his passion. And so was a very like a pure energy with ideas and vision. That is, I think, is very crucial for a leader, for the PI of the group. It's something that I learned from him, and trying to emulate his way to communicate, like his long-term vision, and what is the field, is where the field is going, and actually defining where the field is going. So he was one of those persons that actually the field was following. And this is the reason why with him and also Christian Chenoy, the field of BCI lost like very two big mentors for the vision of the future. And so of course all the others I'm trying, but those were like big loss for the field. And still we are following some of their vision and advices and steps, following their steps for sure. But yeah, what was amazing was really like always learning something new, and it was a great experience. And I'm still copying his attitude also in my lab as well, trying. And of course, I'm not the same because he was really pure energy, a rock star, and I'm not. But yeah, a great experience for me with him.
What was your most memorable experience working with him?
Actually, this moment very, very famous also in the lab. So the first implants of this kind were performed in Pittsburgh, at least in this network of the cortical bionics, with the first participants implanted now 10 years ago with the electrode in the sensory cortex and the motor cortex. And then now they have 3 individuals in Pittsburgh and 2 in Chicago. So in 2020, there was the first implant in Chicago, and Scott Imbrie is also like a good. I invite you to ask him maybe like a podcast, because he's a great person and also very well for communication. He's a BCI participant, but wants to tell his story and his motivation to participate in that. So he was the first participant in Chicago, and Sliman before the implant was, of course, anxious, and it was the first implant. And when we implant this electrode for touch, of course, is very important to place the electrode in the right position in the brain. So to place that on the hand, and in particular on the fingertips, because the youth arrays, these arrays are pretty small. And even like a small shift in the position can change where the sensation that will be provided. Is one of the limitations of this device, of course, where actually the coverage is pretty limited. So you need to place this very precisely on the fingertip. So there is this phase pre-implant pretty long, where actually we are better and better with the techniques, but we need to be sure that the area of the hand and the fingertips are exactly there. So there is this pre-surgical planning, and then there is the surgery. But until, the first week when we start to do stuff, we are not sure about the location, right? If all the predictions were correct or not. So there was this moment where There is actually a video on that as well, where so the first day, one of the first days, so Scott came to the lab for the first phase of recording, and since he's a sensei, so he can feel. So he's a particular case of among the other implanted because he one of the few, or probably the only one, with the still residual sensation. So he can feel sensation, and actually you can play the neurons and see if you are recording something from touch. And this is how the receptive field in monkeys are defined. But so Sleeman was like touching Scott's fingertips and play the neurons at the same time to see if we can hear some response. And then at a certain point, there was like this point where you hear like so the bursting of the activity. And so we were sure that there was fingertip. So we were exactly in the fingertip region. So that was like the moment everybody was happy, smile, and also Scott, everybody. So that was a pretty emotional moment for the group. And Scott will say that the same for sure. And Sleeman was super excited as well. So we were like celebrating. So there were many moments like this. And regarding BCI, I think this was an important one.
Yes, and the first time it's always everyone is anxious. Will it work? Will it not work? Now, I remember we had first epilepsy surgery in the center that we established here in Florida, in Orlando, and that first surgery, how it will go, and everyone is there, everyone is watching, and when it works, everyone is so happy. Yes,
exactly. And I think for him was very important as well because he was the expert of touch. He will say, imagine if in my lab we miss completely the area of touch, and then this would be like very bad, and we cannot do the experiment, and etc. He was very, very expecting this moment to see if everything went well, and at the end everything went well, and it was great. And
Yes, thank you for sharing this. And this brings me to the next question. Usually, people, when they're thinking about those who are paralyzed, they're thinking about their challenges with moving their limbs. Yes. But people usually don't think that the touch or the sense of touch also might be impaired. So, how frequent is the impairment of touch that goes along with the impairment of movement? And in what cases we still might have that residual sensations that you mentioned? And in how many cases we wouldn't have any of that? So, just how persistent is this problem? How big is it? Because I think it doesn't get enough attention or recognition, at least, that it might deserve.
Yes, no, exactly. So starting from the importance of touch, so we know that our manual dexterity that characterize us as humans and also shape in a certain sense our intelligence, because an intelligent behavior, because with the hands and with our fine sense of touch, we can express our art, our fine abilities to assemble the clockwise mechanism, or be a pianist, or so all these abilities are guaranteed by the fact that we have this fine sense of touch that allow us to control our hand in a very fine way and precise and fast. And we know from many cases where this is not possible, but we can also think very easily by a cold day where actually your hand are completely, very cold, and manipulate stuff will be difficult. There are then other proof where if you anesthetize the fingers and you try to do stuff, also you're very bad in that. And then if you go and see a condition where actually you have a sensory loss due to a neuropathy or a lesion of the nerve or degeneration of the fibers, you can see that the motor control is very impaired by that, because vision cannot really replace touch. You're not precise in the same way. It's not suitable to extract tactile information from the environment. And then you're not able to precisely exert the right amount of force to know exactly the configuration of the hand. And so there are many evidences showing how touch is important for our manipulation in particular, and interaction with objects. So in case of spinal cord injury, of course, accordingly, depending by the type of lesion, and here we are talking about cervical spinal cord lesions, here we have the impairment affecting the upper body as well. Of course, in case of a tetraplegia, so like can be complete, you have the completely loss of sensory and motor function at all. And then there are like a big chunk of the spinal cord injuries that are partial and that are affecting immediately the motor function. Then accordingly, where is the lesion? If it's dorsal, ventral, is affecting the motor, the sensory, but they're very different one to the other. And you have the loss of motor or sensory function or both. And this is something that can be evaluated. Of course, in the moment that you lose the motor function, having the sensory are not really useful for your functional activities, no? Because without motor function, you can feel if someone is touching your hand, but you cannot actually interact. But there are many cases where maybe there is some residual sensation, but this is impaired because few fibers are arriving here, but most of them are damaged or are lesions. So you have some sensation, but it's very crude and not specific. So again, you have these problems of not feeling that if something is touching, maybe you can understand that something is touching the hand, but you don't know where, you don't know how strong, you don't feel maybe pain, or you don't feel temperature, only feels very hot or very cold, but not in the middle. So you have very different condition. The point is that in particular in this architecture, this system that we are trying to create, so to having an extracorporeal limb that the patient can control directly, in that moment, even if the subject has some residual touch, to have the ability to control this arm in a dexterous way, we need to add a touch on that. Because if not, only looking at what you are doing, you will not be able to use this device in a very efficient way. So this is the reason why, even if there is a spinal cord injury individual with some residual touch, since we are using an extracorporeal limb, we have to be able to provide the touch also in that.
Yep, it makes complete sense. And restoring touch was the topic of your work for many years already, yes? Can you provide us just with a brief overview of what you've done so far before this new study already with stimulation of the cortex of the brain?
Yes, so I started with working with the peripheral implants, so electrodes implanted into the peripheral nerves, in the somatic nerves, and of amputees, both upper limb and lower limb, where actually we stimulated these nerves accordingly to sensors placed on bionic hand or a bionic foot. And stimulating the nerve, what you can do is, since the nerve is still going to the brain, to the somatosensory cortex, and from the study that also recently has been published, we know that this map of the brain and your body map are stable, are still there. If we stimulate the nerve, even after years of amputation, you can evoke sensation perceived on that, on the hand, on the phantom hand, or on the phantom foot. So actually, we are using what is the residual activity, but since there is no the hand anymore, we are bypassing and go directly in the nerve. But those afferents were normally innervating the hand, so if stimulated, can evoke a sensation on that. So I extensively study how to stimulate these fibers and the periphery to create meaningful, informative sensation for prosthetic hands and feet. And then I move in the field of BCI, and where actually we cannot stimulate the nerves in people with spinal cord injury because the lesion is above, so it's blocking the message to going up. So in this case, we can or target the spinal cord or the nuclei or the thalamus or the cortex or the brain structures. So there is this approach where the idea is to target the somatosensory cortex, the primary somatosensory cortex. Why? For three main reasons. The first one is that is easier to be accessed surgically compared to deeper structures like the thalamus, the cuneate nucleus, or other regions where the signal is passing through. Because here we just need a craniotomy, so to remove the scalp, and then you have already exposed in the posterior sulcus the somatosensory cortex, the primary somatosensory cortex. So it's superficial and can be accessed with the current neurotechnologies available. The second thing is that this part of the brain is actually somatotopically organized. What does it mean? It means that your body is represented on this cortical surface and then on this part of the brain. So different parts are encoding and are processing messages of different parts of your body. So that means that if you find a part of the hand, you will have your hand represented with all your fingers from the thumb to the pinky finger, all represented there. So it's already well separated. And this is, for example, different compared to the nerves when all the fibers are mixed and you need to be very selective in activating one or the other. Here, you just place the electrode in the right position and you can target the different parts. So we have an easier access. We have this somatotopic organization, and finally is large enough to be targeted in the sense that the size of the somatosensory cortex make the interface with our current technology feasible because our technology is millimeters, squares of something, some millimeters, so we can place this and the size is matching. Other regions are very small and compact, so you will need a more selective and small electrode to target that because everything is concentrated in a smaller structure, like the cunate, for example, is much smaller. Everything goes there and then becomes again bigger in your somatosensory cortex. So since it's larger, you can place electrodes in an easier way. So in this way, this was the idea that they had at the beginning to target this area to close the loop of the BCI. And this is what we did in these studies. Even I can tell you more about how we arrived at this study, but this is more or less the background.
Yes, thank you. And you already mentioned some of the similarities and differences, yes, with the stimulation of the peripheral nerves. You would have a bundle of nerves and you need to figure out which one to stimulate and how to do it. Here you have a more clear picture, yes, somatotopic organization. Yes, you stimulate this, you get this finger and this part, and all is more clear. Can you tell more about the difference between the studies that you've done with the peripheral nervous system and now central nervous system? What things you carried from those studies into this new project? And what were the new challenges that maybe you didn't have in your previous projects that you needed to solve?
No, exactly. This is what exactly I started to when I approached this field was exactly you create immediately parallelism, differences, similarities. And first of all, a part of this organization that is different in the brain is also that, as you mentioned, so here we are in a network of neurons, cortical neurons of different types. And in other cases, you have parallel fibers, afferents that are going up. And here, when you stimulate, the network, the entire network is activated, is propagating this. And so the prediction of what you actually can do with the stimulation are a bit more difficult to have because it's not more one-to-one activation or similar to that for the axons. Here you have a more complex network that is also connected with other networks that are the motor networks, the posterior parietal cortex with higher level structures. So the network is pretty complex and it is also, you are quite high in the hierarchy of the processing of touch. No? So if you imagine that you have to send a signal artificially, so from the periphery, you actually send something that was just processed by the mechanoreceptor, and then we are in the nerve. So we are pretty early stage, which was just the mechanoreceptor processing. And then we send the signal up. But if we send in the somatosensory cortex, in theory, what you have to replicate is all the process done up to there. So the cunate, the spinal cord, the nuclei, the thalamus, everything, because I'm not just relays of I'm actually doing some process. There are some important works on that. So the first challenge for us, and actually Sliman, there is like a documentary on 60 Minutes when he actually explained this, that his first idea was, okay, if we have to do this when we stimulate the brain, nothing will happen. I mean, it's so complex that we have to do all this process that it's not possible that if we brutally stimulate this area of the brain, we can elicit anything being like anything informative, anything. And actually, he was wrong, because even with a very simple stimulation of this part of the brain, the subject was perceiving sensation, like on the finger, and able to describe that. So that was actually a thing that we thought was a challenge, but actually was not really a thing. Another like very surprising thing is that when I started to work with this participant, the stimulation in the brain was described more naturally in terms of quality compared to peripheral nerve. That was actually also surprised me for me, like was very surprising, because I was saying, okay, we stimulate the nerve, and then we exploit all the process of information, but somehow the information arrive more corrupted, and then the sensation is more paresthetic, not really natural. Well, if we stimulate the brain directly, the subject is reporting most of the time also electric feelings, but most of the time like touch sensation pretty naturally. So this was also another surprising. And of course, from the level of safety here for the brain stimulation is a bit higher compared to the periphery, because of course you're stimulating the brain, and what you want to avoid is to evoke any type of seizure or any type of a network overactivation, since you are stimulating directly the brain of the subject. So the level of safety and parameter exploration is a kind of more smaller, and you need to be planned carefully every step. Not it was not done. I mean, also for the periphery, but here we are, as you can imagine, another level of invasiveness in terms of stimulation, and also to explore all the type of stimulation that we, biomimetic or not. You remember, here for the brain, we have a little bit more strict exploitation of the technology to encode the sensation. But we managed then to do something as well. But of course, that was a different thing compared to the peripheral nerves.
Yes, and I was very curious about that. Yes, that you were trying to create those naturalistic sensations in your peripheral work with the peripheral system, and you were using different parameters. You found certain things that can create those more naturalistic sensations. How did you apply from that work to your studies of in central nervous system? And how did this finding that actually even a simple stimulation of the somatosensory cortex already feels more natural? Yes, to people, how did this play in all this picture of this biomimetic stimulation?
So this was one of the points, and we actually started in the same way as we did for the periphery. So I have to mention that the BCI, the somatosensory BCI, it's a bit behind the peripheral nerve stimulation in terms of achievement and results, for two main reasons. One is because historically the first implant was done in 2016 in one participant, and then now there are more participants implanted, while for the early 2000s, the peripheral implants for amputees and prosthetics started. So there is a bit of gap historically. And also because in general here we have then also a brain control that is more complex to do than EMG control of the prosthetic, because you have many degrees of freedom here of the elbow, of the wrist, etc. And combine that with touch is still not explore in the same way as all the combination of functional tasks for the periphery. So we are a little bit behind. So as we did it for the periphery, with Lehman also we started, so he had a lot of information, data, and he was the expert on that, on how the brain and this part of the cortex was encoding tactile information. And the first things that we observed was that at the population level, so not at the single neuron level, but both at the population level, the response of neurons in the cortex and in the nerves is not super different. So for example, I'll make you an example. So if I press your finger and then I release, we have these two contact and on and offset of the contact. And this is encoded in the periphery as a first burst of activity when they touch. Then if you stay pressed, there is a bit of activity, but much less. And then when you release, there is another burst of activity. This kind of response is also present in the brain, in the broad matter area 1, so this area of the primary somatosensory cortex that we target with our array, for the same type of stimulus is present these two bars of activity at the population level. Then if you look at the individual neurons are responding slightly different. So, but when we stimulate with our device, we actually activate a group of neurons. So at the population level, we observe this similarity. So this was making us believe that the biomimetic strategies that were working with the periphery maybe could work also with the cortex. So for the biomimetic, there is another study published in 2025. So this year from Taylor, Obbs, and Rob Gaunt, exploring a little bit this biomimetic stimulation for making the sensation more natural, in particular for those channels that are prosthetics, with promising results. But the idea with Sliman that we had was to try to start a little bit simpler, respect to having already the biomimetic source on the biomimetic stimulation for touch in general. We started from trying to combine multi-channel stimulation and multiple channels at the same time with this study with tactile edges and motion, the one that we are talking about today. Trying to understand how the parameters are influencing the perception and how multiple channels may be combined together to encode some tactile feature to give more and more informative feedback to the subject.
Yeah, thank you. You mentioned that for this project, also you used some information from visual cortical prosthetics. Yes. So before we get into the results, I still want to understand more. Yes. So you brought also something from other type of neuroprosthetics into this project. So what was that?
So looking at historically, so for the somatosensory BCI, what was achieved so far was by the same group, by the same people. So what we were able to do was, since the cortex is organized somatoptically, the first point is to encode contact, so where the sensation is felt, in order that if you touch with the prosthetic hand, when you touch with the index, you feel with the index. When you touch with the thumb, you feel with the thumb. And that was pretty straightforward because when you place your multi-channel electrode array, so the youth array in this case, if you change channel and you go from the lateral to the medial part of the cortex, you actually move from the thumb to the pinky because of the somatotopy of the brain. So that was pretty easy to do. And we were able to encode the location and the contact pretty easily. Then, as we did for the periphery, you can map the amplitude of your stimulation and the frequency of stimulation with the perception. So similarly to the periphery, what we observed was higher is the amplitude, higher is the intensity, higher is the frequency, higher is the intensity. So the magnitude of the response was modulated by these two parameters. And with this, it's possible to encode force, for example. So that was actually the state of art of the somatosensory BCI. So able to provide contact and provide a force feedback. That is already enough to do functional tasks, of course. But so with Sliman, we thought, okay, the dexterity of these devices is still very bad. We need to go beyond that. And one of the things is why they're not very good is because the information that we provide is still very basic. When we touch something, we know immediately the shape of the object, the texture, curvature, edges, where to take the temperature, so many different information at the same time that contact and force are not enough. So to do that, we proceed, as I told you, in two ways. One way was, okay, let's see how edges, motion, curvature is encoded by the brain in the neuron. So how these tactile features are encoded in the neuron. And textures, edges, all this type of stuff was extensively studied by Sliman and his team. On the other hand, I started to explore also visocortical prosthetics, because another type of sensory prosthetics that actually started before this in the 60s, 70s, then there was a gap, and now again, different groups in Spain and in Netherlands and in the US, they're doing this. And also Neuralink has a program on visual prosthetics. Actually, it was a similar idea to place an electrode or type of electrode on the V1, so in primary visual cortex, or also other V2, V3, up to V4, but mostly like V1. I was focused on V1 since there are similarities with S1, because we are in S1 and those were in V1. And actually, there were this experiment where when you stimulate in visual cortex, you evoke a phosphane. So this blur contrast image that's with different shape or different sizes when you stimulate from one channel. And I observed that in non-human primates, they showed that they were able to combine these phosphanes in lines and letters. So my intuition was, okay, here in touch, we don't have phosphanes, but when we stimulate from one channel, actually the subject is perceiving one spot here. What happens if we combine this one with the other? So it was pretty trivial idea, nothing like rocket science, but they're saying, okay, let's try to combine this projected field in a meaningful way, in an informative way, to encode some information, for example, about the directionality and the orientation of something touching your finger. To do that, one of the first things that we did was to study if you can add projected field linearly, like if you can really add these things one to another, or when you stimulate with 2 or 3, the combination is some kind of non-linear combination and create another. So the first thing that we observe is that it are actually pretty additive. So when you have this projected field, so the patch of skin that is perceived when you stimulate, you can combine that in shapes. And the subject was reporting to feel an edge with a specific orientation accordingly to the combination that we were doing. And it was pretty remarkable because sometimes we did like some weird, combination, and the subject was actually drawing this perception here as the one predicted by the combination of the projected field. So the first result was actually about the orientation, and we did this in 2 subjects. So what was pretty like surprising, and that was the first kick of the study, because when we observed that it was possible to actually combine these projected fields and have a shape, we had all the ideas to proceed with the study.
That is remarkable. Also, going back to what you said, that there was a different expectation at the beginning, at least by Professor Basmaya, that it will be difficult to evoke any sensations, yes, by stimulating somatosensory cortex. And it also, you were thinking that it might be not as natural as with the peripheral nervous system stimulation. Why did it turn out actually even in opposite? Yes, it was evoking more naturalistic sensations, and it wasn't problematic. Looking now from current perspective back, how do you explain this? What do you think?
Still under debate, so we are still trying to explain that. But one idea that we had was the fact that, so if you imagine that when you stimulate peripherally, you stimulate at the exactly very early stage, and you can imagine that you are inserting with your artificial stimulation pretty noisy signal, right? Because it's not as the mechanoreceptor output, but it's more like an artificial activation. And similarly to, with this, there is like a game where you write a letter or something on the back of the person in front of you. And then the other one is to draw in the front. And at the end, the message is a kind of mess. What we think is similarly that we insert some noisy signal at the periphery, and then you have different synapses, different stages. But this noise is kind of amplifying, and at the end arrive very aberrant paresthetic, and so this is not interpretable. While if you go directly there and you activate the neurons and the correct neurons already, you can evoke maybe a more a meaningful sensation. This is one of the the hypotheses, but we don't know exactly why there is this difference. There's actually one study just put on the meta-archive by the Case Western, Emily Gracic, one participant, both in a single individual, both the peripheral and the cortical, so on the same subject. And also his subject who can actually compare the two said that the cortical stimulation is more natural than the peripheral. I think she also has some theories on that. But then also here the area that we are stimulating is also encoding. There are specialized neurons already responding for specific features. And so this is not the first area of the cortex where the message arrives. So there is area 3D that is in the sulcus, in the central sulcus. So it's difficult to reach. And that is even more at early stage. So it's probably more similar to V1, but it's not accessible. So we go on area 1, Rodman area 1, that is on the surface. So it's accessible with our surgical implant. And stimulating that, we have sensation of different kind, but some of that are also paresthetic, also for the brain. And I don't want to say that everything is natural, but it's just that there are maybe more channel naturally. And it also is described as more natural, but it's still something that we are investigating. But in this case, what's interesting that regarding the type of the individual characteristic of the individual projected field, when you combine that, those are like perceived as a line in some direction. So that was remarkable.
It is. It is absolutely remarkable. And what did the patients report? What were their experience having all those sensations?
They were pretty excited about the fact to perceive something, in general, you have to imagine that they experience when we provide touch is normally without any context. They are there, we stimulate, and they have to describe what they feel. It is actually pretty difficult because it's a kind of similar if you are in the dark and I place something on your hand and just passively because they cannot explore, right? So we stimulate, they perceive something for a few seconds, and then they need to tell what was that. So every time for them, it's not super easy to describe the sensation. And then, of course, you don't want to bias them. So you ask, and then they are open to describe as they want. But then when the sensation becomes more like this, more multi-channel, more with some feature on that, they were able to describe this more clearly. And I think they were excited for that because instead of one point where they have to say something, here there is some feature, something in that. And I think it was pretty cool there. I have some videos on that, but also for motion, because the next step was, again, in my opinion, now posteriori, pretty trivial, but at the time was not, because it was the first time that we thought, okay, if we did we combine this projected field together and you can evoke like a kind of line or the subject can tell if it's a horizontal or vertical line or there is no line or something like that. Let's see if we activate one after the other very quickly. What happened? If something, maybe how the subject will perceive that. And again here, but how quick, how fast we should change channel one from the other. And again here I look to the neuroscience in vision. So there is actually an experiment in vision, pretty I think in the 60s or even before. The way they describe the effect of flashing lights. So if you have two flashing lights and they are alternating, you see two lights. But then if the temporal gap between one and the other is becoming very fast, you start to perceive that is one moving on the other direction. And then again, if it's too short, then you start to perceive two simultaneously. So the first test that we did is to activate two projected fields with different temporal gaps. And with short temporal gaps, the subjects were saying, I feel two touches simultaneously. Then if you start to have a gap under 1 second, 500 milliseconds, the subject will start to perceive one thing moving up. And then if the gap is too much, two taps, alternating, not anymore a continuous motion. So we realized that we can encode a continuous motion on the skin if you have the correct temporal gap between one channel and the other. So we repeated the task of before, but instead of giving lines, we activate these channels with actually different gap. And the subject was able to tell, I feel something moving on my skin in this direction, in this other direction. So I was able to tell both if there was motion or not, and also in which direction. That was pretty cool, because also was something that also was demonstrated with phosphane and visocortical prosthetics for vision. So that parallelism, I was really fascinated by the fact that in both devices, actually in two different fields, with the same paradigm, we're able to encode a similar thing, but in two different domains with the same encoding strategies. This is the reason why recently we have talked more, because the fields before were working separately, but now we are trying to communicate more and see what is working in visual cortical prosthetics and somatosensory cortical prosthetics, and talk more because of that.
Yeah, and how those two fields can help each other. Yes, experiments in one would advance another, and vice versa. Yes, and those main principles of, in general, sensory perceptions, yes, they seem to work in different modalities, the same principles. Mm-hmm. Yeah. Yeah, that's beautiful. So, if you would summarize the goals of the study and then the results that you actually got at the end.
Yes, so the study was aiming to expand the repertoire of artificial touch for somatosensory BCI and trying to leverage the knowledge from non-human primate research and what we know about how the brain is encoding motion and edges and more complex sensory features, and also taking inspiration from visuocortical prosthetics, trying to encode something more complex. So what we achieve is that simultaneous stimulation of multiple channels with the intracortical microstimulation was able to encode sensation of edges and oriented stimuli, tactile stimuli, that the subject was able to recognize. And the sequential activation of these channels were able to encode motion. And then finally, we actually did two more complex experiments. One was to try to encode more complex shapes with the two mechanisms. One was simultaneous activation. So we define some shapes. In this case, we choose letters, but can be any different shape that you have. Encoded that with simultaneous stimulation, more like if you press this on the skin, imagine, or to trace that with the sequential stimulation. So if you have a letter and you trace. And our hypothesis was you have a property of your sense of touch called graphesthesia. So you can read with your sense of touch. So if I trace the letter on your skin, you are able to read what I'm writing, and you are actually much better than compare if I indent the same letter on the skin. Indeed, we found that the subject was able to discriminate the different shapes better if you trace the shape on the cortex as the natural touch. So that was the first comparison of something more functional. And then we did an experiment in closed loop, so with the prosthetic hand. So because the idea was, okay, now that the subject can feel this more complex sensation, let's try with the robotic hand, the robotic arm. But of course, we had some limitation in terms of sensorization, because to give the sensation of motion, you need to be able to track motion on the prosthetic hand. So you need to have sensors for that. So we need electronic skin for that. And so we did an experiment with the brain control robotic arm that was actually used for steering a wheel. So in this case, we encode a sensation of motion, apparent motion of this steering wheel in one or the other direction. And the subject was asked to correct the motion accordingly to this sensation of motion perceived. So this was just a proof of concept to show that the subject was able to control with the brain signal and the robotic arm, and at the same time, you exploit this artificial sensation, this more complex sensation, to do a functional task. And so we proved that was possible. So this was the first idea, and then now we are going through something more complex and maybe textures and something like that.
Yes. So what are your next step now? Now that you started already mentioning something, yes. What's the next step? What we will be hearing about next time you come to our podcast?
It's a meet. I mean, this year I cannot submit because I'm actually the jury chair. I'm honored that this year will not submit, but maybe next year. That is actually was a remarkable result that we are trying to replicate with more subjects, of course, because this was in two subjects, and we want to test also this in all the other subjects. But the idea is to going back to the biomimetic stimulation again and define like something more on the biomimetic studying how the brain is encoding this feature, and use biomimetic stimulation to encode maybe textures and compare that with more classic way to understand that if really the biomimetic theory is valid also here and is a way to, I mean, it is actually what we have to go. and to encode more complex sense of touch. Then there is another challenge for the cortical somatosensory prosthetics that is actually S1 M1 communication. So when we stimulate on S1, so differently compared to the periphery, since here we have a network, where actually you stimulate on S1, you have a lot of activation on M1. So where we take the signal for controlling the hand. It is not just artifact. It's not that if we stimulate in S1, we create this artifact that then can ruin our motor decoder or can affect our ability to control the hand. But it's actually modifying the activity in M1 because it's information. So if M1 is receiving information from S1 accordingly to the tactile event that are happening. So we need to create motor decoders that are able to synergistically take this information in to create a functional bidirectional BCI for touch, because that is a challenge that is there for us that we want to do this type of bidirectional prosthetics, and was actually not really a problem for the periphery because we were recording from the muscles, and then even if you stimulate the nerve, you don't create a lot of artifact or a lot of activation in the muscle. No, you can actually decode that. But for the brain, since everything is there, you have a lot of modulation induced by S1 that we need to take into account.
Yes. Yeah, I will be looking forward towards the updates. And would you say were the main innovations by the project that won the award? Mhm.
Yes. For sure, the main innovation is about the level, the richness achieved in terms of artificial touch, because these type of features have not been encoded in any type of neuroprosthetics in general, considering peripheral, spinal, and cortical. Before was not possible to achieve something like that. And maybe I'm interested to go back in the periphery and see if I can do something with agile motion for the peripheral stimulation, but this was not possible. And I think also I really like this comparison with visocortical prosthetics that we mentioned, because I think this is the study that can really put the connection between the two fields. And there are many things that they try and they fail, or they succeed, and vice versa. It will be very useful to go there, look carefully if some of the things that they managed to achieve is valid also here, because this is a clear example where actually we can translate some result in another field. We can do maybe the same thing with the other things that we achieve easily, or we achieve because we invest more time in that, and we can see if it can work also for visocortical prosthetics.
Yes, and speaking about that, you mentioned that visual prosthetics was actually it was one of the first that people really wanted to create. Yes, but then because of the complexity of it, yes, it was abandoned. People switched to cochlear implants, which was a very successful project. But now it looks like the visual neuroprosthetics is on the rise again. So with that said, what is the current picture in the neuroprosthetics world? What is again being popular? What are the main areas of research, of work? What are the major trends? And what can we expect in the near future in this field? Just taking more global perspective now.
Sure. No, exactly. So first of all, it is a field that is growing incredibly, thanks to the investments, and also I think also the public interest also push on that. And personally, it's cool. So I think more and more I feel students that are super excited to work in this field. So this is in general a good point for this type of neuroprosthetic field. With the coming of new electrodes and interface, I feel that this can be really something that can change a bit the possibilities that we have, because up to now, a few years ago, in particular for stimulation, but also for recording, very few were the electrodes FDA approved, and CE mark even less, but FDA approved for this type of research or this type of clinical intervention. So now there are more companies developing novel type of interfacing with the brain that can open up the possibility to do more in human research and in human trials. And exactly as you mentioned, the visual cortical prosthetic, maybe years before, considering also the technology available to interface the brain was not enough to achieve that, but with a technology with high resolution, high channels count, higher channel counts, maybe go deeper as what Neuralink is proposing, or something that can target a different area. So I feel that more tools are available, so this can expand the usage. For sure, the motor BCI historically is the one that started as a control of a cursor to give what they call now digital independence to the user. So and be able to control your computer, your smartphone, etc. I think is something that can soon help patients, and what Neuralink, Synchron, and other are proposing as one of the main points, no, for the. I mean, I'm saying for actually the clinical benefit for the patient, not for a research tool and devices that are available. And speech neuroprosthesis, I think, is, as you know, and also the BCI Award, is mentioning this multiple times, in the sense that there is growing in terms of a beat per second and words per second, that we can to have a very natural way to communicate with the natural pace and that we have now. In inner speech, they show so very, very interesting applications of speech neuroprosthetics. The somatosensory is, of course, I think the next step necessary for the motor to be really to move on something physical in the physical world and control robotic arms and hand and interact with that. And I think would be the field of more cognitive neuroprosthetics. I feel is also very interesting and to look, because it's interfacing with other area of the brain and trying to communicate. Because at the end, I think if we summarize, the application are in the able to decode information from the brain and use that for something, or to communicate and encode information that can be for vision, that can be for touch, but how to communicate back information right into the brain information. So I think there are more and more algorithms. There are people following more AI-based approach, more like black box, but able to provide information. But I feel I'm more from trying to understand the mechanisms of how the brain is working and trying to apply that, and maybe then AI-based mechanism, but after that. But also the increase and the growth of AI methods is connected with that. If we think of large language model for speech, for example, or others. So I think there are many opportunities.
Yes, yes, I can see that the field is exploding. We have several teams already coming up with neuroprosthetics, yes, which is absolutely amazing. And speaking about that, I listened to a lecture of one of, I think, wonderful professors on the topic of, in general, human sensory perception. And the thing that he mentioned that in theory, the possibility of having this matrix-like experience, like in the movie Matrix, yes, when the reality is being created around us by stimulating our nervous system and evoking all the sensations that are around us, including visual, auditory, and of course somatosensory, that theoretically it is possible. So we really we can live in a virtual world that will be as real as ours, but technically it's still not possible. So how close are we getting with the development, including your work, yes, to the time or to the possibility when this completely naturalistically feeling a virtual world will become possible? What do you think?
I feel honestly, it's we are far from that. I hope as well, because I don't want to leave it. I want to live the real life, not the virtual one, but this personal. But yeah, so what I want to mention is that it's true that our subjects are already using virtual reality settings, and they control the virtual limb, and they can feel from the virtual limb while they are doing stuff. So we don't use always the physical robot, because it's burning energy and is also noisy and can break. So when it's needed, we use it. But if we want to have something that we can run in virtual reality, we can easily also place objects and parameterize that. So technically, the subject is already using an arm in virtual reality, and they can experience sensation from virtual reality. And it's actually also very interesting why they describe the experience in virtual reality compared to the physical world, where it's actually the physical world where they see the robot shake the hand of a person and they can feel that. That is a very different experience compared in the virtual reality. But the point is that we are still really lacking of technology to interface at the level that is needed, in my opinion, for encoding. We can see in visocortical prosthesis, but also in our case where we can, yes, evoke sensation on the fingertip only because we can cover only that part and with a limited amount of information. So our touch is still an impoverished version, is still a pale representation of the natural touch. So this is the reason why I feel that the way is still long. There's still, in our case, for example, proprioception is still missing or other important information. So in my opinion, we are still far from that. But the goal is that, is right to replicate and really communicate and find a way to communicate with the brain. That is, my belief will be the game changer in terms of treating diseases and in general to connect us with the external devices.
Thank you. And another question is related to what became available to us recently. Until this time, I think a general opinion was that when the limb gets amputated, there is reorganization that is happening in the brain, and there is a takeover of the adjacent areas of the brain area that was responsible for that limb. And there is a longitudinal study that is showing that that might not be really the case. So I think this research got a lot of attention recently. So I would like you to comment on that. So what are these recent findings and what do they mean? And for the field of neuroprosthetics and for the field of BCIs in general.
Yeah, no, that's a very good point because actually it was. So I feel that Anders Schoen is the first author of the study published this year, like this month actually, last week in Nature Neuroscience with the Tamar Macking, University of Cambridge, and actually it's for the field of neuroprosthetic. I think is a very important study for many reasons. First of all, because it's something that in the field, at least in our field, was already something that we had in mind, and but never really demonstrated at this level and with this rigor. Because if we implanted, and I participated in clinical trial both for the periphery, so amputees, leg, arms, and spinal cord in the brain, and we got the implant in different parts of the nervous system to restore sensation. People amputated maybe the few months before up to 20 years, 30 years before. And also spinal cord injury the year before or 30 years, 40 years before. But in all these cases, when we stimulated this part of the brain or the nerves, the subject immediately perceived sensation where was supposed to perceive, like hand, foot, never like face, lips, or something. No? So that was already like a kind of proof or indirect proof of the fact that there was not really invasion of area, like the area was still there, ready to receive information. And so the hypothesis was that and also Sleeman was also a promoter of this theory, that thanks to the evidence that he also like saw in the field of neuroprosthetics, where actually after the critical period, so this period when you are young, when there is like more plasticity, then after the critical period, in particular, your body maps and the sensory maps needs to be stable and reliable, because you don't want to have these changes that I'm sitting here, imagine, and then if I stay hours here with my elbow, then the brain representation of the elbow becomes larger than the hand. So you don't want that. You want to have this stable map that you refer to every time that you interact with the environment that is very stable. But of course, there were like studies showing this type of reorganization. So it's a kind of trick, to find, I mean, in particular, if you're not a scientist that is studying exactly that. But recently, there was also a very nice paper published by Tamar Macking and John Krakauer on eLife describing this theory against cortical reorganization. where they describe that the studies in that showing this type of organization was just a misinterpretation of the data, or could be, in the sense that one example was the fact that if you have an area of the brain that is receiving major inputs from one area and then also other inputs from the other areas, when you cut this input because of the amputation or because of the lesion, and you look at this area now is activated by these small other inputs. And so you misinterpret that this area is now a face area, but was already connected with the face. It's just that now the inputs are arriving only from the face. So it seems activated during the face, but before was not the case. So there is like upregulation of function that was already present before amputation, that this was a kind of interpretation. But they explain very well in this review or in this like opinion. And with this study that they published now, they have the final proof where they have people before amputation, where they measure the map in a very detailed way of the brain with fMRI. And then after amputation, at different time point, months up to years, and they show that this map remains stable. So this is an important, in particular for neuroprosthetics, because that means that we can recruit and implant people regardless the time, no? Because even if you're amputated in 23 years, you can have a prosthetic like that. You can have an implant. There is no problem related to the reorganization. So this is for the neuroprosthetics. And for pain is also about the theories of the treatment, because the theory is saying this maladaptive plasticity, this invasion can be something that is affecting how you perceive and related to phantom pain, where maybe the theory is more with the peripheral inputs or something like that are now probably more reasonable or something like that. So this is where, I mean, this study is important because it's touching, I think, on different points and also revising the literature from before.
Yes, but those, I think, absolutely amazing new developments and demonstrations, that support the development of neuroprosthetics. Based on all these developments that you mentioned, what's going on right now in the field, what do you think opportunities that are opening up for people who want to join the field, where they can find themselves?
Sure, yeah, exactly. So first of all, in a small way, we as a lab, we are growing. We are recruiting students, PhD and master students, etc. And since it's a multidisciplinary field, we take students and to approach this problem of the different point of view, from the more on the algorithm, more on the hardware, more on the clinical part, and then implementation. I feel that now there are more companies also where actually you can have an experience both in Europe and in US in the field of neuroprosthetics that can be valuable, and they are growing as well with searching more and more people. And I feel that also there is this diverse diverse environment in terms of approaches. There are some that go intracortical, epicortical for the same goal, or you have neuroprosthetic and BCI for different aspects. So not only for, as was before, for control the cursor of the screen, that was the BCI field, at least invasive, because non-invasive much more. But for now invasive, there are many more like techniques, and also groups studying the different aspects and different type of diseases. So I feel that there are more opportunities both in academia and industry.
And industry is something that people are really trying to get in right now. It becomes very popular. And what would be your advice, yes, for people who want to enter the field? I'm sure people are contacting you with all kinds of questions. I just had a person who came for a consultation, and we even had a little chat, about how many messages do you receive, because he sent a message to you, and that's how the conversation started. He didn't receive the reply yet because it was just recently, but I told him, don't worry, I'm sure Giacomo gets at least 10 messages like this per day. He says, no, not 10. I'm sure he receives hundreds of messages a day. So
Not from many, not from many. No.
yes.
Sorry.
So, but you are receiving at least, many questions. Yes. So what is that, the most common questions that you hear? Maybe you can answer it right here so that they wouldn't need to contact you about it.
Yeah, no, exactly. So in general, so as I said, that it's very nice that the students are really attracted by this field. They also have to consider that there is a lot of background that you need to learn, and there are very different aspects, and every part is fundamental altogether. Of course, everyone wants to come here, work with the patient, and do the everything, but of course, is like piece by piece, and someone has to take care about one part, some other on the other part, and then put together. So of course, there are maybe small steps, and this is something difficult, I think, for the students to try to understand that is a small step towards something. You cannot jump there immediately, but I feel that is important to show your interest, your genuine interest in the field, and of course be open to try also something that can be challenging for you or maybe out of your comfort zone, but you is also an opportunity to learn something new. And of course cannot be only the thing that you study, but since this is a very multidisciplinary, if you are from make an example, like a computer scientist, and you come here to write algorithms, but you don't know anything about the brain, you can do it. But of course, as a student, is something that, since here we do stuff related also for the disease touching neuroscience, medicine, engineering, you need to have an overview of everything and also study this type of stuff that maybe is not completely of your interest, but in particular as a student, you need to be flexible to learn and to be a sponge, and try to learn as much as possible to then specialize on one specific thing. This is my suggestion.
Thank you so much, Giacomo. Again, it's always a pleasure. Was it how different it was for you to submit the BCI Award application this year compared with the last year? So what did you learn that maybe you can share with our listeners about the submission that came with the experience for you?
Yeah, so I feel that the application per se was very similar in the sense that it's pretty straightforward, and the goal is to explain the project. Of course, the committee and the jury is a jury of experts, so it's a bit easier as a researcher because you can write actually a project summary in detail, and because actually you are evaluated and on the detail, because they are experts, so they know the field is not about the project, but the novelty about the idea in this field and why is deserve an award, So you need to convey the message about the novelty compared in particular to the state of art. So this is something that I feel we improve. And also I spend a little bit more time on making the videos, explanatory videos about the experiment, because there is this possibility to have a short film on that. So we recorded a little bit more video about the experiment, more professionally, and also in a more like explanatory, and then of the things that we were doing. And I think that is also sign of something that is novel, and if you can show that actually, and you can see that, is actually easier. So it's a kind of nice that I started. This is not the first. That was not the first time of the submission. Exactly, started with the nominee, and then like the third place, and then first place. So it was actually rising. So it was nice. It was very nice, and so I will consider again. Of course, as I said before, now I'm in the jury, and with Christoph, we found also other great members. So I'm really proud of the jury this year, diverse and coming from all over the world, from different expertise, and I'm really curious to see the application of this year. And so I now I'm the other side, so I know the difficulty to convey that. So I will look to something very innovative and interesting.
Yes, absolutely. I'm also looking forward to it. And thank you again for coming to our podcast, looking to the future podcast with you. And maybe we can do one with you and the patients that you worked with. And as we're ending our podcast, is there anything you would like to share with our listeners? Maybe something that I didn't ask, but you still want to mention?
Yeah. Maybe just one thing about since you mentioned the participants.
Yes.
I want to thank the participants. I highlight this because they are the pioneers, and they are also part of the BCI Pioneers Coalition. That is a coalition founded by one of the ex-participant, so a participant that finished his experience in a trial, so that he's now the leader of this coalition. They are wonderful people. We call themselves superheroes because they have different powers, because they say, oh, okay, I have a speech neuroprosthesis, the other has the motor neuroprosthesis. So they meet in events, and it's very nice. And for students, I really suggest to watch some of their videos or events, because they can have really give you the impression of the experience of someone using that, and also they are part of the group. They're the real pioneers in this research, and without them, nothing would be possible. So really, I want to thank them for donating their time and life to this, and they're coming every day to run sessions. So it is an incredible effort for the future, for science. They do this for the next generation. So
for the future. Yes, absolutely. And I'm so glad to see this coalition because they can support each other. I think that's so important. And also support future participants. I think, yes, I'm so glad to see that. And I'm looking forward to doing a podcast with them. So thank you again, Giacomo. All the best. And we will see you in our future podcast then. All the best with your studies. Thank you.
Thank you, Milena.
Dear Neurocareers podcast listeners, thank you for joining me and my incredible guests on this exciting journey into careers in neuroscience and neurotechnologies. I hope you've been inspired by the stories of those turning groundbreaking ideas into impactful realities. If you are looking for more guidance on advancing your neurocareer, book a consultation with me, Dr. K, your podcast host and neurocareers coach at the Institute of Neuroapproaches, the only career service dedicated specifically to the needs of professionals in neurotech. Let's take the next step toward your neurocareer success together. In addition to free consultations, the Institute of Neuroapproaches offers a variety of services designed to help you thrive in neurotech. First, professional development planning. We help you create a tailored career development plan, identify knowledge gaps, and equip you with the tools needed to achieve your goals in neuroscience and neurotechnologies. Second, resume and cover letter review. Get expert feedback on how to craft documents that are specifically designed for job opportunities in the neurotech field. Third, interview preparation. Sharpen your interview skills through mock interviews and targeted feedback, including soft skills, hard skills in neurotech, BCIs, e.g., MEG, live coding, and presentation preparation. 4. Networking and job search strategy. Learn how to effectively network and uncover job opportunities while standing out in the competitive neurotech job market. 5. Negotiation and salary advice. Receive expert guidance on negotiating salaries and benefits and determining a fair salary range for your desired position. All this and more can be found at www.neuroapproaches.org. Again, www.neuroapproaches.org. So, what are you waiting for? Let's navigate the path to success in the world of neurocareers together and turn the impossible into possible. We're looking forward to working with you.